Okay, Assalamualaikum dan selamat sejahtera to all my students. Alright, so let's not wasting our time. So let's continue our discussion on the 12.2 variation in humans and the second subtopic in the chapter 12 variation. So let's take a look on the learning standards that you have to cover for today. Okay, number one, you are able relate variation to human inheritance. Relate variation to human inheritance. And number two, conduct an experiment to study continuous and discontinuous variation in humans. Alright, so that are referred to the learning standards for today. Okay, let's go to the relation of variation to human inheritance. So we already learned human inheritance in the chapter 11. Alright, so what are the relation between the variation and the human inheritance? Okay, human trait. Right, so do you still remember what is a trait? Trait is the refer to the variation of a specific characteristics. Yeah? For example, if the characteristic is a height, so the trait for the height are tall and a dwarf, or tall and short, right? So human trait is controlled by a pair of alleles, by a pair of alleles in which an allele can be dominant or recessive. The dominant trait is observed when both dominant alleles are present or when one dominant allele is paired with a recessive allele. Okay, that is referred to the dominant trait. Dominant trait is controlled by the dominant alleles that present in the genotype. Right? So whether they are homozygous or heterozygous. Right? So that is a dominant trait. What about recessive trait? Recessive trait is only shown when both recessive alleles are present. Right? So this genetic information cause variation in humans. Okay, so you have to remember because of the presence of the dominant alleles and recessive alleles. Yeah? So this genetic combination, genetic information cause the variation in the humans. So these are referred to the example of characteristic and traits in the humans. Okay, the characteristic, for example, height. So the traits in the human, okay, dominant trait shown is tall and short is the recessive. What type of hair? Dominant is curly hair and recessive is straight hair. Okay, tendency to use hands. Right-handed is a dominant trait. Left-handed is a recessive trait. Presence of the dimples. Yeah, so dominant trait is presence of the dimple, and the absence of dimple is referred to the recessive trait. Ability to roll the tongue. Can roll the tongue is a dominant. Recessive is cannot roll the tongue. Types of earlobes. Okay, dominant trait is free earlobes. Okay, and recessive trait is attached earlobes. Okay, so here refer to the diagram of the different characteristics in the humans. Presence of the dimples refer to the characteristics. Type of hair, type of hair, whether straight or curly, right? Ability to roll the tongue, type of the earlobe. So all of these refer to the different characteristics in the humans, right? Okay, now let's go to the third subtopic in the variation, chapter 12 variation, okay, which is 12.3 mutation, right? Okay, mutation. Okay, so these are referred to the learning standards for the mutation, yeah, right? Okay, so you have to able to understand, yeah, describe the terms of the mutation, mutant, right? And then number two, describe the types of the mutation. They refer to the physical agents, chemical agents, and also biological agents. And number 3, describe with examples types of the mutation. That refer to the two types of the mutation, which are gene mutation and chromosomal mutation. And last one, relate mutations of somatic cells and gametes to the variation. Okay, so these are referred to the learning standard for the 12.3 mutation. Okay, so what is a mutation? Right, mutation is a spontaneous and random change of genetic material, namely DNA, right, of the cell in an organism. Okay, so in other words, so mutation is occur spontaneous and also random change, cause a random change of the DNA, of the DNA. Alright, so a substance which causes mutation or increases the mutation rate to a dangerous level is called mutation. Okay, second terms that you have to know is mutagen. What is a mutagen? Refer to the a substance or agent that cause mutation. Which cause mutation, that is mutagen. Alright, so the first term just now is mutation. So what is a mutation? It is a spontaneous and random change of the DNA of the cell, right? Okay, and then we go to the next. Mutation occurs spontaneously in natural condition. Right, so mutation occurs spontaneously. So mutasi ini berlaku secara spontan, tidak dikawal, tidak dikawal. In natural condition. So new genetic material produced by mutation is called mutant. Okay, this is referred to the third terms that you have to know. Mutant. So what is a mutant? Mutant refers to the new genetic material produced by mutation. New genetic material produced by mutation. Okay, so a mutant can exist as mutant gene, mutant cell, mutant organelle, or also mutant individual. Alright, so that is referred to the mutant. Mutant can exist whether as a mutant gene, mutant cell, mutant organelle, or the mutant individual. Right, so we go to the types of the mutation. So what is a mutation? Mutation refers to a substance that causes mutation. Okay, so mutation can be divided into three types of agent, namely physical agent, chemical agent, and also biological agent. Right, so we go to the physical agent first. Okay, physical agent that cause mutations are ultraviolet from the sun, UV ray, ultraviolet ray from the sun, and then ionizing rays such as X-ray, alpha ray, beta ray, gamma ray, and so on. Right, so that are referred to the ionizing rays. Okay, that are physical agent. Okay, then we go to the chemical agent. Chemical agent refer to the chemical substance, right? So first, carcinogen in cigarette smoke. Carcinogen, cigarette smoke. What is the carcinogen? Agent that cause cancer in the cigarette smoke, such as tar. Nitrogen oxide, and so on. Right. So then, food preservatives, also chemical agent that cause mutation. Formaldehyde. Formaldehyde. We can find it in the Clorox, right, or any any cleaning agent, right. And then benzene, right? Benzene refer to the also the chemical substance, right. So all of these are chemical agent. And then we go to the biological agent. Biological agent that cause mutations are virus or the bacteria. Alright, so these are referred to the three types of the mutation. They are physical agent, chemical agent and also biological agent. Now, okay, we go to the types of the mutation. Okay, just now types of the mutation. Now we go to the types of the mutations. So there are two types of the mutation. Okay, what are they? Gene mutation. Okay, gene mutation and chromosomal mutation. Gene mutation and chromosomal mutation. Okay, again, what is a mutation? You have to know what is a mutation. Mutation is a spontaneous and random change of the DNA of the cell, right? So, mutation can occur in two types, whether gene mutation or chromosomal mutation. Okay, we go first to the gene mutation. Yeah, gene mutation. Okay, gene mutation occurs when there is a change in nucleotide base sequence of a gene. Right, so change in nucleotide. Nucleotide. Do you still remember what is a nucleotide? Nucleotide refers to the basic unit of the nucleic acid. So the nucleotide consists of what? Consists of the three components. Do you still remember what are they? Yeah, they are ribose sugar, pentose sugar, phosphate, group, and also nitrogenous base. Right. So when there is a change in a nucleotide base sequence, it will cause gene mutation. Yeah, it will cause gene mutation. So gene mutation is also known as point mutation. Yeah, point mutation. The change alters the genetic code that is used to synthesize amino acid. Therefore, there will be a change in protein structure, and this new protein cannot function. Right? Okay, so you have to know that when the mutation occur, when the change, when the genetic code is changed, so what happened? So it will produce a different structure of the protein. So it will produce a different structure of the protein, and this protein cannot function. Alright, okay. So the gene mutation occurs by base substitution, base deletion, and base insertion. So there are three types of the gene mutation. What are they? Base substitution, base deletion, base insertion. Okay, later we are going to discuss one by one. Okay, let's take a look on this diagram first. So this is referred to the gene mutation. Okay, what happened here? So what is this? CTAGAT. So these are referred to the nitrogenous base. Yeah, nitrogenous base. So this is referred to the DNA, right? DNA. So this is referred to the nucleotide. So this is referred to the nitrogenous base. So what happened here when the mutation occur? So at the C here, C refer to the, what is C? Do you still remember the nitrogenous base of the C? Okay, C is cytosine. So cytosine is changed with the, or C is substitute with the T. T refer to the thymine, right? So it will cause the changes of the gene, changes of the gene, of the DNA. Okay, so these are referred to the example of the mutation or the gene mutation, the changes of the nucleotide in a gene. Okay, we go to the three types of the gene mutation. Okay, what are they? They are referred to the base substitution, base insertion, and base deletion. Okay, base substitution. Substitution ini refer to the replacement. Penggantian base-nya, nitrogenous base-nya digantikan dengan base yang lain. Okay, we go to the diagram here. So this is referred to the DNA. So the words here refer to the nitrogenous base. So in the sequence, okay, what can we see from the sequence here? So this is referred to the base T. Okay, base T, nitrogenous T, refer to the thymine. Thymine is replaced or is substituted with the G, yeah, guanine, with the guanine. Okay, and then what happened here? Okay, we can see here. So no more T here. So T is replaced or substitute with the G. And of course, it will produce the new amino acid. Yeah, the original sequence will produce the glue. Yeah? Okay, GLU, right? So it will produce the GLU amino acid here. But when the mutation occur, when the base substitution occur, so it will produce a different amino acid. Yeah, so when it produces a different amino acid, it will produce, it will synthesize a different protein structure. Alright, so this protein structure cannot function. Okay, so that is the first type of the gene mutation, base substitution, whereby the base, the base nitrogenous base is substituted with a different base. Alright, and then we go to the second type of the gene mutation. What is it? Base insertion. Base insert. Base is inserted here. So a new nitrogenous base is inserted in the sequence. Alright, okay, and then so the original one is no adenine, no adenine base here, but in the new sequence, okay, so the base is inserted here. Base insert, and then it will cause the production of the new amino acid here. Okay, this is the original one, and this is the refer to the new amino acid. So new amino acid are synthesized and produce the new protein structure, which is also cannot function. Okay, and then we go to the third types of the genes mutation, which is base deletion. The base is deleted. The base is deleted. So what happened here? The G here, okay. So G, the guanine base here is deleted. It's deleted or being removed, right? And then what happened here? So it will produce the new amino acid. Okay, because the sequence is already changed. The sequence of the base is already changed, so it will produce the new amino acid. Okay, so all of these mutation, of all of these types of the gene mutation will produce a different or new amino acid, and then it will cause the production of the different structure of the protein. When the different structure of the protein, that protein cannot function. Right? Okay, so that is referred to the gene mutation. Okay, so the gene mutation causes genetic disease such as thalassemia, cystic fibrosis, sickle cell anemia, albinism, and also hemophilia. Thalassemia, cystic fibrosis, and sickle cell anemia, albinism, and hemophilia. So this refers to the thalassemia. Thalassemia. Alright, okay, we already discussed thalassemia in the inheritance a little bit. So thalassemia occurs because of the abnormal hemoglobin. So it will cause the red blood cell cannot transport the oxygen efficiently. Right, and then this is referred to the cystic fibrosis. Cystic fibrosis cause the production of the mucus in the lungs, and that will cause damage to the lung. Right. Now we go to the types of gene mutation. Example of the genetic disease based on the type of the gene mutation. So base substitution, example of disease sickle cell anemia. Base insertion, okay, example of disease is cystic fibrosis. The damage of the lung because of the excessive production of the mucus. And then base deletion. Base deletion, example of disease is thalassemia. Thalassemia. Okay, so this is referred to the sickle cell anemia. Sickle cell anemia. Sickle cell anemia is caused by a gene responsible for the synthesis of hemoglobin. Right? So what happened? The hemoglobin of the red blood cell is abnormal, and it cannot function efficiently. It cannot transport the oxygen. It cannot transport the oxygen. So the red blood cells of a patient are in the shape of a crescent. Yeah, the red blood cells. So as we can see from the diagram here, the red blood cell of the patient is in the shape of the crescent. Ya, berbentuk bulan sabit. Ya, kamu boleh nampak di sini bentuk dia macam bulan sabit. So this is referred to the normal red blood cell. So this is referred to the sickle red blood cell, in the sickle cell anemia patient. So this is because the red blood cells are not properly formed. Properly formed because of the abnormal hemoglobin. So some of the red blood cells are normal, whereas the rest are crescent shape. So that is a sickle cell anemia because of the base substitution. Substitution that occur. It is one of the type of the gene mutation. Okay, we go to the second one, albinism. Albinism is one of the disease that caused by the gene mutation. So albinism, an individual who experience albinism is an albino. Okay, so this person is albino. Because he experienced or he have albinism. Okay, albinism is due to the mutation of a gene which is responsible for producing pigments of skin, hair, and eyes. Alright, so what happened in the albinism? The mutation occur of the gene, mutation occur, and it will cause no production of melanin. Production of the melanin refer to the pigment of the skin. Okay, so this albino, the pigment of the melanin are not produced. In the albino, the pigment of the melanin is not produced because of the gene mutation occur. Okay, so these are referred to the albinism. Albinism. Okay, next we go to the chromosomal mutation. Chromosomal mutation. So chromosomal mutation involve changes to chromosomal structure or changes to the chromosomal number. Changes to the chromosomal structure or changes to the chromosomal number. Alright. Okay, chromosomal mutation can change the characteristics of an organism. So this will cause the change the characteristic of an organism. The changes in chromosomal structure involve changes to the gene sequence in a chromosome. Okay, so you have to remember that chromosomal mutation will involve the changes to the chromosomal structure or any changes that occur to the chromosomal number. Alright, okay, that is referred to the gene mutation. Chromosomal mutation. So the structural change causes abnormality to the chromosome, which is known as chromosomal aberration. Chromosomal aberration refers to the abnormality to the chromosome. So types of the chromosomal aberration include deletion, duplication, inversion, and also translocation. Okay, so these are referred to the types of the chromosomal aberration. What is a chromosomal aberration? It is referred to the abnormality to the chromosome, because of the deletion, duplication, inversion, and translocation. Okay, let's go to the types of the chromosomal aberration just now. Alright. So deletion, duplication, inversion, and also translocation. So what happened in the deletion? Yeah, in the deletion, a part or a segment of chromosome is deleted or lost. So means lost a few genes. Yeah, it lost a few genes because a part or a segment of the chromosome. So original chromosome, okay, as we can look here, there is a six segment A, B, C, D, E, F. Okay, so when the mutation occur, what happened? The segment B is deleted here, and it will produce a mutant chromosome with no B segment. Okay, that is referred to the deletion. Okay, duplication, a part of a chromosome is copied, which causes repetition of a gene sequence. Alright, so original segment, there is a six segment A, B, C, D, E, F in the chromosome. And then what happened when the mutation occurs? There is a segment of the chromosome is copied, yeah, copy, and it will produce a mutant chromosome with a double or with a duplicate of the segment here. Yeah, so there is a two segment B here, okay, because of the duplication occur. Okay, and then inverse. Yeah, inverse. Okay, inverse. A segment of chromosome is reverse. Yeah, reverse end to end at 180 degrees Celsius. Then rejoin. Yeah, the change in the DNA sequence of the chromosome. Okay, so what happened here? The original segment is A, B, C, D, E, F here. So what happened during the mutation? The segment of the chromosome is reverse. Yeah, reverse and to the end. Yeah, and then so A here go to the F, F here go to the A. Right? So the segment of the chromosome is reverse. Yeah, reverse to the 180 degree Celsius, and it will produce a mutant chromosome. Yeah, mutant chromosome here. Alright, so actually it's not the A here, it is the any segment. Yeah, any segment. Yeah, okay, so any segment, maybe it's just a part, or maybe or all the segment, maybe one or two segment, right? That is referred to the inversion. Okay, and then we go to the translocation. Okay, translocation, part of the chromosome is cut, then joined to another non-homologous chromosome. Yeah, parts of the chromosome is cut, then joined to another non-homologous chromosome. Okay, so this is referred to the chromosome. Yeah, which is referred to the non-homologous. So non-homologous chromosome. So what we can see from here, so this refer to the A, B, C segment D, E, F, right? And then what happened? Right, what happened? So these segments, okay, is cut, and then this segment join. Yeah, is cut and join to another non-homologous chromosome. Yeah, okay, it's cut and change, right? So DEF will go to the here, and then JKL will go to the here. All right, that is referred to the translocation. So at last it will produce a mutant chromosome. Okay, so these are referred to the types of the chromosomal aberration that are involved in the chromosomal mutation. So what is a chromosome mutation? Refer to the changes, yeah, in the structure of the chromosome, and also changes in the number of the chromosome. So all of these are referred to the changes on the structure of the chromosomes. Okay, a change in chromosomal number. Okay, now we go to the chromosomal mutation that involve the change in the number, chromosomal number, right? Okay, a change in chromosomal number cause a diploid organism to lose one or more chromosomes or gain, yeah, whether it loss or gain, one or more chromosome. Okay, the change occurs when homologous chromosomes fail to separate during anaphase 1 in meiosis, or sister chromatids fail to separate during anaphase 2 in meiosis. So actually this also we already discussed in the chapter 11. Alright, I hope no problem on that, right? Okay, so this is probably because the normal spindle fibers fail to form during meiosis. So this phenomenon is known as non-disjunction. Yeah, non-disjunction. Okay, what is a non-disjunction? When the homologous chromosomes fail to separate during anaphase 1 in meiosis, or the sister chromatids fail to separate during anaphase 2 also in the meiosis. That is referred to the non-disjunction, and it causes abnormality in the number of chromosomes in a gamete, right? Okay, the gamete may lose one or more chromosome or gain one or more chromosome. Yeah, the gamete maybe have one less number of the chromosome, or maybe it will has extra chromosomal number. Okay, so these are referred to the non-disjunction that occur during anaphase 1, and also non-disjunction that occur during anaphase 2. So these are referred to the non-disjunction in homologous chromosome during anaphase 1. Okay, non-disjunction in homologous chromosome. And at the end, it will produce the gamete either has extra chromosome, or the gamete has less chromosome. Alright, so in non-disjunction during meiosis 2, okay, what happened here? So normal disjunction in homologous chromosome during anaphase 1. So this is repeated normal disjunction, whereby the homologous chromosomes separate to the opposite poles that occur normally. And then, right, so in meiosis 2, okay, what happened here? The non-disjunction in sister chromatids during anaphase 2, okay, whereby the sister chromatids do not separate during anaphase 2. And last, it will produce the different gametes with the different number of the chromosomes. So you can see here, so this gamete will carry extra chromosome. This gamete will carry less chromosome. Okay, so all of this process of the non-disjunction during meiosis can cause abnormality to the number of the chromosomes. Abnormality in the number of chromosomes can be due to non-disjunction during spermatogenesis or oogenesis. So we already discussed this now. Non-disjunction that occur during meiosis in formation of the sperm and also formation of the secondary oocyte. So if non-disjunction occur during these two processes, it will cause abnormality in the number of the chromosomes. So fertilization that involves abnormal gametes will produce a zygote that develops into an individual with abnormal characteristics. Okay, so the individual experience change in the phenotype. So it will cause a change in the phenotype. Okay, so we take a look on the figure here. Right. So if non-disjunction occur of the sex chromosome during oogenesis, during oogenesis, the formation of the secondary oocyte. So what happens if non-disjunction of sex chromosomes occur? An abnormal secondary oocyte is fertilized by a normal sperm. Right? Okay, so an abnormal secondary oocyte is fertilized by a normal sperm. Okay, so we can see here, so this is referred to the secondary oocyte. Secondary oocyte with no chromosome. Yeah, with no chromosome inside here. So with no sex chromosome. Yeah. Okay. Here, secondary oocyte that have extra sex chromosome. Yeah, secondary with extra X chromosome with two X chromosome. So if fertilization occur, okay, we take a look on this one. So this is referred to the gamete that carry none of the sex chromosomes. Yeah? So what happened here? Fertilization occur. So it will produce the zygote with XO. XO. So only one sex chromosome is present in the zygote. So it will cause the Turner syndrome, female, which is female Turner syndrome. So if the secondary oocyte fertilized with the sperm that carry Y, Y chromosomes, so it will produce a zygote with the OY. So this will cause the zygote to die. Okay. And what happened to the secondary oocyte that carry extra X chromosome? Yeah. So if it fertilized with the sperm, so it will produce a zygote with XXY. Yeah, sex chromosome that has XXY. So this will cause Klinefelter syndrome in male. Yeah, Klinefelter syndrome male. Right? And what happened here? Yeah, secondary oocyte here with an extra chromosome fertilized with the sperm that carry X chromosome will produce a zygote with a triple X. So this refers to the triple X syndrome female. Okay, so all of these occur when non-distension of sex chromosome during oogenesis. Okay, and then we go to the non-distension of sex chromosome during spermatogenesis, during the formation of the sperm. Okay, a normal secondary oocyte is fertilized by an abnormal sperm. Okay, we can see here the gamete produced, okay, the sperm produced is abnormal. Yeah, abnormal. Why abnormal? Because the sperm here do not carry any sex chromosome. Okay, but the sperm here carry both sex chromosome, X and Y. So if fertilization occur between the sperm and the secondary oocyte, here normal secondary oocyte. So what happens? It produce a zygote that has sex chromosome with XO. Also will cause Turner syndrome in female. Yeah, Turner syndrome female. Okay. If the sperm that carry X and Y chromosome, fertilized with the normal secondary oocyte, so it will produce a zygote that has XXY sex chromosome, with extra X chromosome. So this also will cause Klinefelter syndrome in male. Klinefelter syndrome in male. Okay, so all of these diseases are caused by the mutation of the chromosomal number. Alright? Okay, so I hope you can understand. Okay, let's take a look on the characteristics of disease caused by the chromosome mutation. Alright, so example of the disease here, Down syndrome. Yeah, this is referred to the abnormalities in autosomes, and abnormalities in the autosomes. Okay, so the disease here refer to the Down syndrome. Okay, what happened in the Down syndrome? The chromosomal number of the Down syndrome patient is 47. 2N, okay, with extra one chromosome. Has 21 chromosomes. In the 21st pair, he or she has three chromosome on the 21st pair. Means they have extra chromosome on the 21st pair. So these individuals, okay, so this individual, either male or female, yeah, has slant eyes, matanya sepet, flat nose, protruding tongue, broad forehead, and is usually mentally retarded. Alright, so these are referred to the Down syndrome individual. Alright, so make sure you can remember the characteristics of the Down syndrome. Okay, next we go to the disease which is Cri-du-Chat syndrome. Yeah, Cri-du-Chat syndrome. Yeah, so the structure of chromosome changes due to a partial deletion on the short arm of chromosome number 5. Yeah, okay, what happened here? What happened? The mutation occur that cause the structure changes. Yeah, the structure changes on the chromosome number 5. Yeah, chromosome number 5. Okay, so what are the characteristics of the Cri-du-Chat syndrome? Okay, the cry of affected infants is similar to that of a meowing kitten. Right? Okay, the cry of affected infants are similar to that of a meowing kitten. Affected individual shows delayed development with mental and physical retardation. So most patients die during childhood. Okay, so what happened to the Cri-du-Chat syndrome individual? So the usually infant when they cry, they will produce a sound that's similar to the meowing kitten. Ya, bila bayi ni menangis, dia akan berbunyi seperti bunyi kucing kucing mengiau. Alright, okay. Right, so that is Cri-du-Chat syndrome. Okay, now we go to the abnormalities in sex chromosome. Abnormalities in sex chromosome. So now involve the sex chromosome. Okay, Klinefelter syndrome. Okay, Klinefelter syndrome, the number of the chromosome is 47. So extra one chromosome. So 44 plus XXY. Yeah, 44 plus XXY. So extra X chromosome in the sex chromosome. So what is the characteristics of the Klinefelter syndrome man? Yeah? A sterile man with small testes that fail to produce sperms. Okay, possess voice and chest similar to those of a woman, has long legs and hands. Okay, that is referred to the characteristics of the Klinefelter syndrome individual. Okay, second one, Jacob syndrome. Okay, Jacob syndrome also have 47 chromosomes. Yeah, 44 plus XYY. Now this person has extra Y chromosome. Yeah, extra Y chromosome. 44 plus XYY. Okay, that is referred to the Jacob syndrome. Okay, male who is taller than normal. Yeah, taller than normal and has problem with pimples on his face. Slow in acquiring speech and has learning disabilities. Possess weak muscle. Okay, it is hypotonia. Okay, that is referred to the Jacob syndrome, whereby this person has extra Y chromosome on the sex chromosome. Okay, then Turner syndrome. Turner syndrome occur in the female, right? So what happened here? The number of the chromosome of the individual is 45 chromosome. Yeah, 44 plus X2. The missing of the one of the sex chromosome. Alright. So what is the characteristics of the female? Sterile female with a lack of secondary female characteristics. Yes, sterile female with lack of secondary female characteristics, undeveloped breasts and ovaries, okay, web-like neck and low IQ. Okay, that is the characteristics of the Turner syndrome. Okay, make sure you can know the disease that caused because of the abnormalities in the autosomes and also abnormalities in the sex chromosome. Right, okay, last one. Yeah, last one here. Mutation of somatic cells and gamete. Okay, mutation can occur in somatic cells and gamete to produce variation in a population. So what are the difference? Okay, what are the difference between the mutation in somatic cell and the gamete? Okay, mutation in somatic cells involve somatic cells such as skin cell and eye cell. Okay, involve somatic cell. Mutation in the gamete involve germ cell that produce the gamete. Yeah, involve germ cells. Yeah, germ cell refer to the parent cell. Yeah, germ cell that produce gamete. Okay, and then the mutation in somatic cell cannot be inherited by the next generation, but mutation in the gamete can be inherited by the next generation. Mutation in somatic cell disease is present only in the individual with the mutation. But in the mutation in gamete, disease is present in the individual with the mutation and is also inherited by his or her descendant. Alright? Okay, and then example of the mutation in somatic cells refer to the disease related to the nervous system. Yeah, example of the mutation in the gamete, all inherited disease, yeah, such as thalassemia, sickle cell anemia, right? All the disease, yeah, albinism, yeah, all the Down syndrome, okay, Klinefelter syndrome, Turner, and so on, right? So all of these are inherited disease, alright. Okay, I think that's all for now, right? Okay. I hope you can understand all the subtopic for the 12.2 and 12.3 about the mutation. Right. So please read again and again. Do your own notes, do your mind map or concept map until you do really understand. Alright, okay, that's all for now. Okay, take care everyone. Thank you.